Good morning. Hello. My name is Julia Bernier, and I'm an investigator and researcher within the Cancer Research Program of the Research Institute of McGill University Health Centre in Montreal, Canada. And firstly, I'd like to thank you for the opportunity to give you a brief overview of what I'm doing in Montreal and how it relates to ocular melanoma and some of the exciting projects that we have. I think it's great that this symposium is going forward, even despite the current global situation. And so I thank you again for the invitation. Our university and research institute is really facilitating translational and clinical research because our new hospital and Glen site allows for our hospitals and research institutes to all be under one roof in one site. So at the new Glen site super hospital in Montreal, we have our clinical hospitals, pediatric and adult facilities. We have our state-of-the-art research institute, a dedicated cancer center, as well as supporting infrastructure for platforms and scientific research. And this really allows us to do bench to bedside translational research all under one roof with our clinical and scientific colleagues. My research, which is within the Cancer Research Program, focuses on molecular biology and cancer biology. And in particular, I have an interest in ocular melanoma. And so that's what I'm going to talk to you a little bit about today and some of the exciting projects that we have. My research focus is really under three main umbrellas. One of the things that we're doing is trying to understand the molecular mechanisms underlying metastatic disease, and in particular, trying to understand how cells go from a primary location very specifically to secondary organs, so what we term site-specific metastasis. And as you know, ocular melanoma has a remarkable tropism to the liver. And so one of the things that we do in the lab is try to understand the communication between tumor cells and the microenvironment of the liver organ. The second thing that we really focus on, and I'm going to talk a little bit about today, is designing clinically feasible approaches to monitor disease course through liquid biopsy. And some of you may have heard the term liquid biopsy and be familiar with this approach. I will briefly go into it, and I think you will have had the chance to also hear about this earlier in the symposium on a talk on circulating tumor cells. And I think this is a really exciting field, a new direction that we're going in that could have big impact for patient care. And lastly, we work to uncover the role of how the molecules that we find within liquid biopsy that I'll discuss actually play a very active role or have mediate the progression of tumors and metastatic disease. And in particular there, we really focus on therapeutic targets. Recently, we've teamed up with an engineering group that is going to help us design therapeutic vehicles for us to be able to bring potentially some exciting molecules closer to clinical testing. So what are liquid biopsies? Again, I think you will have heard a little bit about this, but this is a really exciting new field. And simply put, liquid biopsies is the use of biofluids, such as the blood, to sample tumor or disease molecules. So in the area of cancer, we really talk about our ability to use biofluids to sample, isolate, characterize, and study tumor disseminated molecules. And liquid biopsies aren't only in the blood. We can use saliva and urine and ascites, and in the eye, we can use aqueous fluid and vitreous. But the most commonly studied is the blood. And it is so because it is very minimally invasive to take a blood draw and to isolate and characterize disseminated tumor molecules. There's three main types of analytes that we talk about. One is circulating tumor cells, which I think you will have heard about, which are tumor cells that detach from the primary tumor and enter in the circulation. And they can be identified because tumor cells have differences in density and morphology and size than normal cells of the body, as well as cell surface molecules that can be used to recognize them. There's also circulating DNA and other types of nucleic acids, and this is a big focus of my research. These are small fragments of DNA that are released by tumor cells, enter the circulation, and we can identify them based on their genetic characteristics, which are reflective of the tumors that they stem from. and extracellular vesicles, which are small membrane-divided vesicles that reflect their cell of origin. I like to think of them as mini cells or mini representations of the tumor cells that they derive from. And in recent years, really exciting research has shown that extracellular vesicles are likely one of the communicators or mediators of progression, metastasis, communicating between the cells of the microenvironment where tumors will grow. And I think what's really important about liquid biopsy is that it gives us a lot of insight into tumor heterogeneity. We know that tumors are not homogeneous. They have different clonal populations, cells with different histopathological and different genetic characteristics. And those different characteristics will also dictate differences in behavior. So by being able to sample a representation of all of this, I think is really important, and it gives us insight into not only the cell populations within that primary tumor, but if there's more than one tumor, if there's micrometastatic disease. So I think it's a really powerful way to study disease in a robust way. And there are many applications of this kind of approach. As I said, it's a non-invasive biomarker for early detection of disease. It allows us to monitor patients non-invasively over time, which I think is a really crucial gap right now in our clinical monitoring, that we cannot biopsy patients over and over again. So we are unable to really sample disease over time. And I think this is a really important feature of liquid biopsy, to detect disease, relapse, or metastasis prior to it being clinically seen. To evaluate early treatment response, one of the things that we're doing a lot in the lab now is looking to see if we can use circulating tumor DNA and other types of liquid biopsy to evaluate treatment response in preclinical modeling. And I think we have compelling evidence to suggest that that is the case. And ultimately, really, the goal is to modify treatment according to the molecular changes that we see. And so if we're able to track and monitor patients closely, then we're able to modify treatment in a really personalized way. And I think this is really the basis for personalized medicine. So one of the types of work that we do, and I wanted to just give a little brief snippet of one of our studies, is an analysis of liquid biopsy looking at circulating tumor DNA from a preclinical to a clinical study with a very consistent methodology. And the aim here really is to see if we can detect tumors and their progression in ocular melanoma through sequential non-invasive biopsies, and importantly, in a cost-feasible way. So what we did is we've conducted a really thorough in vitro cell culture analysis to detect DNA that comes from tumor cells and DNA that's embedded within those vesicles that I mentioned, also coming from tumor cells, to see if they reflect the genetic components of those of that DNA reflects the DNA of the tumor cells and of the primary tumor from which they come. And what we've found is that this is a really good model to study disease and to study circulating tumor DNA. So then we moved into a clinical study where we've enrolled close to 50 patients now. I think we're at 45 with uveal melanoma and some with nevi. And we were looking to see if we could detect circulating tumor DNA, and if circulating tumor DNA would inform us on disease, on response to treatment, and as a predictor of disease progression. And I think here we have some really exciting data to suggest that this could be a really important biomarker. We're doing this at the McGill University Health Center, and we've partnered with the Centre Hospitalier de l'Université de Montréal, which is our French hospital in Montreal. And now we've opened up to include the Cleveland Clinic, Arun Singh, and Wills Eye Hospital, Carol Shields, and now São Paulo Medical School as well with Rubens Belford. And I think it really exemplifies how coming together we can enroll more patients, we can get more data, and it can be really more significant and make more progress. This is a rare disease, so by coming together, we can really make more of an impact with our research. So we can use this type of methodology and approach to answer lots of questions. We can use this approach to answer clinical questions, such as, can we use liquid biopsy and circulating tumor DNA to monitor disease course, in terms of tumor burden, progression, metastasis, and I think importantly, at what stage is this clinically significant? Is this going to be more important for detection of disease or for monitoring or as a biomarker of treatment efficacy? And maybe this is even going to help us stratify patients to particular treatments based on what we see in liquid biopsy. But importantly, and I'm a basic researcher, I'm a cancer biologist, we can also understand the disease better. So can we identify circulating tumor DNA in all of our uveal melanoma patients? And does this give us some insight into mutational profile and the genetic component of their disease? So can we genomically characterize these tumors through a liquid biopsy, which I think would be very powerful? And to do so, we need to compare the genomics of tumor tissue to that of the circulating material. And that's why I'm excited to see other researchers also looking at liquid biopsies, be it circulating tumor cells or circulating tumor DNA, because I think together we can get some exciting data. So that was a brief kind of introduction into what we're doing. But I think in something like uveal melanoma, we really need to come together and see that it's a multifactorial approach. I'm excited to hear about the research being done by colleagues all over the world that can help bring our clinical diagnosis, our histopathological correlations and characterization, molecular genotyping, biomarker studies, and liquid biopsy all together so that we can come up with more personalized treatment decisions to better treat and monitor our patients with uveal melanoma. So with that, acknowledge the people in my lab. These wonderful faces that you see here are the talented members of the lab that I have an opportunity and privilege to work with every day. You see there my lab address, webpage, and my email address. And thank all the people all the sources of funding that have helped us to accomplish what we've accomplished over the last few years since I started this laboratory, and to continue to make progress in the ocular melanoma and cancer biology fields. Thanks very much.
Hello, this is Dr. William Harbour of the Bascom Palmer Eye Institute and the Sylvester Comprehensive Cancer Center of the University of Miami. I want to thank CureOM and the Melanoma Research Foundation for the opportunity to speak today. These are my disclosures, and these are the highlights of what I will be discussing in this talk. First, uveal melanoma is made up of several subtypes based on their gene expression profile, their mutations, their chromosomal changes, and these are related to their risk of metastasis. So in the most simplified manner, we can divide them into class 1 and class 2 based on their gene expression profile. Class 1 tumors can further be divided into class 1A and class 1B. The class 1As have a very minimal risk of metastasis, whereas the class 1Bs have a more intermediate risk. The 1As tend to be associated with mutations in EIF1AX, whereas the 1Bs tend to have mutations in SF3B1, a splicing factor. Class 2 tumors are very strongly associated with inactivating mutations in BAP1, located on chromosome 3, an association with loss of the other copy of chromosome 3. We can think of this in terms of genetic evolution of the genomic landscape in uveal melanoma. A presumed normal uveal melanocyte within the choroid or the ciliary body or the iris undergoes a mutation in a gene in the GQ family, most commonly GNAQ or GNA11. This typically leads to limited growth, after which the tumor arrests and stops growing and becomes what we would call a nevus. Most of the time, it never progresses beyond this point unless further genomic aberrations are acquired, such as the EIF1AX mutation, in which case the lesion remains class 1. They start off as class 1 and remain class 1 if they develop an EIF1AX mutation, which has a minimal risk of metastasis. If they develop the SF3B1 mutation, again, they remain class 1. but they acquire somewhat higher risk of metastasis. If they instead undergo mutation of BAP1 and loss of the other copy of chromosome 3, they develop the class 2 expression profile and a much higher risk of metastasis. Here I want to focus on this class 2 group with the BAP1 mutations because we know these patients have a very high risk of metastasis at the time their primary eye tumor is diagnosed if they undergo a biopsy and gene expression profiling so that we know that they're in this high-risk group. We would like to develop a therapy that we could give these patients in the adjuvant or preventative setting rather than simply waiting until they develop metastatic disease. However, there is not a known drug that will target BAP1 mutant tumors. Therefore, we undertook a drug screen, which was just published this month in Molecular Cancer Research, in which we initially did an in vitro drug screen based on transcriptional changes caused by BAP1 loss. These compounds were then screened for a secondary screen in frog embryos for efficacy and toxicity. And then a final lead candidate was tested in a preclinical mouse model using human uveal melanoma cells. And this just shows the lead candidate quizzinostat, which is an HDAC inhibitor, rescuing the malformed phenotype caused by BAP1 loss in the embryo, but causing no toxicity. And then here we are in the mouse model. We have two BAP1 mutant cell lines on the left, MM28 and MP46. Quizzinostat significantly blunted the growth of these tumors, whereas it had little effect on the MP41 BAP1 wild type tumor cells. Based on this preclinical data, we are working with the company that licensed quizzinostat to develop a clinical trial for patients with class 2 uveal melanoma in the adjuvant setting. And this would be a phase 2 synthetic control arm design, and you'll be hearing more about that in the future. Next, I want to turn to the tumor microenvironment and checkpoint immunotherapy. As you will know, in cutaneous metastatic melanoma, checkpoint immunotherapy has been transformational in the care of these patients, with many patients responding. And yet, sadly, we're not seeing that response rate in uveal melanoma. We're not quite sure why. To address this question and to look further into the tumor microenvironment in uveal melanoma, we performed the first single-cell sequencing experiments in uveal melanoma using 8 primary tumors and 3 metastatic tumors. And here we see almost 60,000 cells from these tumors that are plotted together so that we can see the overall view of what's happening in the tumor microenvironment. And first, you will note that there are understandably and expectedly a lot of tumor cells, which are depicted in mostly in blue and red, but there are a lot of other cells that are not tumor cells here. And we'll explore this further in the next slides. What's interesting is these non-neoplastic cells, mostly immune cells, they're different between class 1 and class 2 tumors. So it's not just the tumor cells themselves that are different between class 1 and class 2, but the immune cells are also different between these tumor types, suggesting that the genomic aberrations associated with class 1 and class 2 tumors interacts with the microenvironment in different ways to create different immune microenvironments. If we look just at the overall composition of tumors, going from the primary class 1s on the left over to the metastases on the right, we see an interesting pattern. The low-grade class 1 primary tumors are relatively simple. They have some non-neoplastic immune cells, but not a lot. The class 2 cells have more. They're more complex than the class 1 primary cells, tumors. But the metastases are even more complex. This suggests ongoing evolution, not only of the tumor genome, but of the immune microenvironment during tumor progression. Next, we wanted to look further at tumor evolution, and we used for this copy number variation or chromosomal gains and losses. And if you look at our bulk data from a previous publication on the left, this is a relatively simple pattern that we see. But in comparison, look at the pattern on the right that we see using single cell sequencing. What this means is that even though the major genomic aberrations, the copy number gains and losses, and the major mutations that we see recurrently in most uveal melanomas, these tend to happen very early and are present in most or all tumor cells. However, when you look at higher resolution, there is ongoing evolution at the cellular level that goes on beyond that initial burst of genomic aberrations. Most of these are not clonally expanding in a recurrent manner. So we would refer to this as neutral or undirected evolution because you don't see the same pattern over and over across tumors. Another way of looking at this is an evolutionary tree. If we look at the bulk sequencing that we previously published, these trees are very bare, they're very simple. But if we look at single cell sequencing data, we see these much more complicated trees. Now, all of the major genomic aberrations are still present in the trunk of the tree, so to speak, meaning that they occur very early and are present in most or all tumor cells. But then there are these ongoing small branches that are occurring, most of which never evolve, but some of which can provide a growth advantage and lead to further metastatic progression. Now, this is an interesting case here where we have a class 1 tumor that does not have a loss of chromosome 3 or a BAP1 mutation in all of the tumor cells, but in one of the small branches, it first underwent loss of chromosome 3q and then loss of chromosome 3p. So this tumor, if it had not been treated, might have progressed on to a class 2 tumor. If we look at the immune microenvironment, here are those same 11 tumors, but only showing the immune cells. You see just how complex this is. This has really never been reported before. We've only really known about that there were T cells and macrophages in uveal melanomas, but look at the complexity of this immune microenvironment. We can look at the clonal expansion of the B and T cells, and surprisingly, even though these tumors are immunologically cold, so to speak, and they don't respond well to immunotherapy in general, they can mount at least a limited immune response with some clonal expansion. So this gives us hope that if we can understand this system better, we might be able to induce an immune response. If we look at the checkpoint markers that are commonly targeted in cutaneous melanoma, CTLA4 and PD1, these are not very highly expressed. Instead, it's LAG3, a different checkpoint marker that is highly expressed in uveal melanomas, both primary and metastatic. So based on this, we have initiated a clinical trial with Bristol Myers Squibb using their LAG3 inhibitor and PD1 inhibitor here at the Sylvester Cancer Center in association with my colleagues, Jose Lutsky and Lynn Fun. So if we put all this together, yes, we do have this genomic aberrations that occur early, and we would call these directed mutations and copy number changes. These are highly recurrent. They occur early in a punctuated burst during tumor evolution, and they are very canonical. On the other hand, these later changes are undirected or neutral in terms of their evolution. They can occasionally randomly lead to a growth advantage, but they're undirected or neutral in their evolutionary pattern. But and then 8q, people frequently talk about 8q as being a driver in uveal melanoma. We don't really see it that way because you can get 8q gain in both class 1 and class 2 tumors. It's just that the class 2 tumors tend to gain a lot more copies of 8q. And we view 8q as sort of putting fuel on the fire. It doesn't direct the outcome, but it can worsen the outcome once that trajectory has been determined. And we also have to overlay the immune microenvironment now. And we would hypothesize at this point that the class 1 tumors do a better job at, the patients with class 1 tumors do a better job at suppressing metastasis because of the immune response to the tumors, whereas the class 2 tumors seem to be able to hold the immune response at bay and even suppress the immune cells from performing their job. So in the closing moments, I just want to mention the Collaborative Ocular Oncology group, which is an evidence-based, multi-center, prospectively oriented group that performs trials in patients with uveal melanoma and eventually other eye cancers as well. This is the group that developed the gene expression profile that's now clinically available. We've just recently concluded the second study, and these were the investigators in that study. We enrolled over 1,600 patients in a little over 2 years to look at the interaction between gene expression profile, PRAME, next-gen sequencing panel for mutations, and copy number variations. We're seeking to develop a single integrated precision medicine instrument, a prognostic tool based on a single needle biopsy that will allow us to stratify patients for surveillance, to enroll patients in adjuvant therapy, as we have discussed here, and to choose the choice of therapy based on their genomic aberrations. And we've now expanded the Collaborative Ocular Oncology group to include key leaders in medical oncology with special expertise in uveal melanoma. This group is being led by Dr. Jose Lusky here at University of Miami Sylvester Cancer Center, as well as Rich Carvajal at Columbia and Takami Sato at Thomas Jefferson. These will be evidence-based therapies that we will test in signal-seeking, rapidly accruing adjuvant and metastatic trials across geographically distributed centers in the United States. I would like to acknowledge my sources of funding, including CureOM, and I want to thank those in my laboratory who have contributed to this work and to others who have worked closely with us. Thank you.
Hello, my name is Professor Sarah Copeland.
And I am Dr. Helen Callery.
And together we lead the Liverpool Ocular Oncology Research Group based at the University of Liverpool in the UK. Thank you very much for giving us the opportunity to give you an overview and a flavour of the different areas of research we're undertaking in uveal melanoma. Essentially, our research encompasses a range of angles when trying to understand uveal melanoma etiology, its biology, its genetics, how metastases arise, the metastatic niche, and also the application of various in vitro models and the use of animal models in improving therapeutics for uveal melanoma. One silver lining of the pandemic's lockdown has enabled us to tie up lots of loose ends, completing projects and writing papers. Together, Helen and I have published over 16 papers in the last month of 2020, and we'd like to take you through some of these.
So the first paper we're going to look at looks at the genetics of uveal melanoma and the incorporation of these results into prognostication for patient management. So Liverpool is the only centre of the 4 specialist ocular oncology centres in the UK to provide routine genetic testing for patients and to incorporate this into the prognostication for patient management. We currently undertake molecular genetic testing using chromosome copy number alterations, in particular chromosomes 3 and 8, and incorporate this into a prognostic algorithm called LUMPO. We now know from the wealth of literature that has been published over the last 2 to 5 years that copy number alterations are accompanied by key somatic mutations in uveal melanoma specimens, and these need to also be taken into consideration when determining patient prognostication. So the aim of this study was to design a targeted next-generation sequencing panel that looked at both copy number alterations and mutations in routine clinical samples. So we initially took 14 uveal melanomas where we had previous TCGA data and copy number data, and we examined these using two panels. The first of these was the adjuvant SureSelect panel, which uses hybrid capture technology, and the second was the Illumina TSCA panel, which is a PCR-based technology. It was clear from this initial study that the adjuvant SureSelect panel by far outperformed the Illumina panel, both in terms of copy number failures and the number of reads mapped. So we moved forward this panel to examine 117 uveal melanoma, including the 14 previously examined. And what we were able to show was that we could process both fresh frozen formalin fixed paraffin embedded material as well as small intraocular biopsies before and after irradiation and successfully profile these using this next generation sequencing panel. The samples could be subdivided into those that were disomy 3. We had 55 samples that were classified as such, and also 59 samples that were then classified as monosomy 3 or isodisomy 3. What we were able to see from this classification is that the majority of the monosomy 3, isodisomy 3 samples were BAP1 mutant, as we would expect, and also that the majority of the disomy 3 samples had either an IFAX or an SF3B1 mutation, which again is what was expected. Interestingly, 2 monosomy 3 uveal melanoma that were wild type for BAP1 also harboured an SF3B1 mutation, and 2 disomy 3 uveal melanomas harboured mutations in both SF3B1 and IFAX. We also noticed novel frameshift deletions in SF3B1 IFAX, and as well as a PLCB4 mutation that was outside of the hotspot region on exon 20. As has been demonstrated by ourselves and others in the literature, this study also showed that those patients that do worse have a BAP1 mutation. Disomy 3 patients with SF3B1 mutations also do badly, perhaps with the late onset of metastatic disease, and extra copies of chromosome 8q are also associated with a poorer outcome. We are confident, therefore, that this next generation sequencing panel can be moved into routine clinical practice, and this is something that we're now currently developing with our NHS colleagues.
Moving on to another study, we used advanced techniques, including single-cell sorting, to examine the immune profile of primary and metastatic uveal melanomas. So Carlos, one of our senior postdocs, examined the microenvironment of 5 metastatic uveal melanomas and 4 primary uveal melanomas using a range of techniques, including CyTOF, nanostring immunoprofiling, digital spatial profiling, and then applied a range of bioinformatic tools to better understand what happens to the immune response when there is loss of BAP1 within the uveal melanoma cells. To take you through some of the results then, application of the pan-cancer immune profile panel, which is composed of 755 genes, this led to this grouping of the immune genes in both the primary and metastatic tumours. Furthermore, it was interesting to see that when comparing the metastases with the primary tumours, there was concordance for the most part with these genes. When then examining the publicly available TCGA data with respect to the primary uveal melanomas, it could be seen that there was upregulation of several genes associated with a suppressive immune environment when there was BAP1 loss. And these genes are highlighted in the image on the right. When then looking at our samples, it could be demonstrated and confirmed that the major reactive cells within uveal melanomas are CD8 positive T cells and also the M2 macrophage. And the M2 macrophages highly express certain markers which include CD74 and MIF. When we looked at these metastases histologically and using immunohistochemistry, we could demonstrate that in many of them, particularly the more so-called mature metastases, were surrounded by collagenous walls, and that within the metastases, you had these M2 macrophages, but out with the collagenous walls were the CD8 positive T cells. So this was this immune exclusion, which was quite apparent in these metastases and could therefore explain the non-response of immunotherapy in most metastatic uveal melanoma patients. So we wanted to take this work further, and therefore we accessed 40 additional uveal melanoma metastases to the liver and applied nanostring transcriptomics and immunohistochemistry to further understanding of the microenvironment of metastatic uveal melanoma, and also to determine what proteins were upregulated by the tumor cells in the hope that they may be targetable. These 40 metastatic uveal melanomas were examined histologically and using immunohistochemistry and subdivided into cold, altered, and hot tumors with respect to their immune infiltrates. Most of them were either cold or intermediate tumors with excluded T cells and the main macrophage being of M2 type. When applying the transcriptomics, we had 3 arms to the study. First of all, we examined the metastases compared to the background control non-tumorous liver. We subdivided the metastases into those which were positive for BAP1 on immunohistochemistry or negative. And then in some of the cases, we had the opportunity to look at differing metastases scattered across differing lobes and therefore compare the transcriptomics between each of the metastases. In some, there was an upregulation of 10 genes in the metastases compared to the background liver. There were differing transcriptomic profiles between some of the metastases. And on immunohistochemistry, there was overexpression of 7 proteins, which included, interestingly, DSUP4, CD44, IFR4, BCL2, CD146, IGF-1R, and PRAME. So this work is of importance because it has identified new potential druggable targets in metastatic uveal melanoma.
So we've been working on developing in vitro models that will bridge the gap between 2D monolayer cultures and animal studies and clinical trials. So the use of 3D in vitro models is increasingly recognised as more physiologically relevant than 2D monolayer cultures when examining both tumour biology and also the response to drugs. So we can either use monocultures with just the tumour cells themselves, or we can develop more complex spheroids that combine multiple cell types. However, 3D culture is not without its challenges, and optimization of the methods and also the endpoint assays is essential in order to take this work forward. So in this publication, we looked at standardizing some of our laboratory methods for generating these 3D spheroids using both cell lines and primary uveal melanoma samples from our biobank. So you can see here a panel of the 6 uveal melanoma cell lines that we tested. These are routinely used by a large number of labs across the world. The 6 cell lines were of differing genetic characteristics, as were the 9 primary uveal melanomas we tested. And all 6 cell lines form spheroids, but you can see that they were of varying size and compactness, with some forming very small, very compact spheroids, and others forming quite large, loose spheroids. And this was something that we also saw with the 6 primary uveal melanoma samples that successfully formed spheroids. The actual phenotype of these was very, very similar to the cell lines. We also observed that the primary uveal melanoma spheroids were composed of melanA positive proliferating melanocytes, and interestingly had admixed macrophages, which, as we've heard from our previous studies, are also very important in terms of the tumour microenvironment. And this is particularly interesting going forward, and is something that we will aim to develop further. And we can also mention that the primary uveal melanoma spheroids, by the time that we use them in the assays, were genetically identical to the original sample tumour. We then went on to look at 2 uveal melanoma cell lines in terms of drug drug testing, we use the 92.1 cell line, which is derived from a primary uveal melanoma, and the MM66 cell line, which is derived from a metastatic lesion. And we tested two agents, doxorubicin, because it has inherent autofluorescence and we can see how it penetrates into the spheroid, and also selumetinib as an agent that has previously been used in clinical trials for patients with uveal melanoma. And we assessed both drug penetration and toxicity after 48 hours. So we used a range of endpoints. We used a viability assay called 3D cell titer glow, but we also concentrated visually on imaging the spheroids, examining what they looked like at various time points, looking at the compactness of these spheroids and actually their area. And I think this is something excuse me, very important to concentrate on when looking at the assays and the endpoints that you measure with regard to drug efficacy. What we could see was that drugs showed varying penetration into the cell line spheroids. The doxorubicin seemed to pass into the spheroid core, while selumetinib had an effect largely on the peripheral cells, affecting proliferation of the outer cells, but seemingly a viable central core remained. Both drugs, however, did cause a dose-dependent reduction in the viability of the 3D spheroid cells. And so this system is something that we're now moving forward to developing more complex systems and developing the assays that we use for drug testing further.
Another project that we looked at was the application of digital pathology and artificial intelligence for the interpretation of immunohistochemical stains in uveal melanoma. All uveal melanoma samples that are processed in our histopathology laboratory are stained for BAP1. We and others have demonstrated the usefulness of the BAP1 immunohistochemical stain as a surrogate marker for the mutational status of the uveal melanoma cells. In this project, we piloted a deep learning model whereby the model predicted, on the basis of the H&E stain only, whether the tumour cells expressed BAP1 on immunohistochemistry. This work was undertaken by our PhD student Hongren, who applied the model to a well-phenotyped set of uveal melanomas, using this training set and then applying it to a test set using a variety of mathematical models, including ResNet, and then producing a ROC curves which demonstrated the accuracy of the predictions of the deep learning model. We plan to expand this out in a multi-collaborative centre study. I won't have time to go through the other projects that we've been able to write up. However, I'd just like to highlight two. One was a very good review written by Karen Orton on microRNAs in uveal melanoma. The second is a very interesting study looking at uveal melanocyte embryology. This was undertaken in a collaboration with Professor Paul McMenamin, who is a professor of anatomy at Monash University, together with melanocyte biologists at the Beatson Institute in Glasgow. This provides us further insight into what happens when melanoblasts and melanocytes are migrating into the choroid, and thereby provides us with a better understanding of the underlying biology of uveal melanocytes. If you would like to read further about our work, we have a website, and the address is there. Our publications are uploaded, and if you look at via the paper publication tab there on the left, you'll be able to see all of our different publications per year. I'd just like to say that Helen and I celebrated our 50th co-authored paper during the COVID pandemic lockdown, and we're more than happy to take any questions or comments regarding our papers. Here are our addresses, and of course we'd like to thank all of those agencies and charities who have funded our work, our postdocs and our students. Thank you very much.